Morning, everyone, and welcome to the first heat of the 10th annual University of Manitoba's 2022 3-minute thesis competition. Today's heat is being recorded and is being broadcast live on the University of Manitoba YouTube channel. I'll begin by acknowledging that the University of Manitoba campuses are located on the original lands of the Anishinaabe, Cree, Ojibwe, Dakota, and Dene peoples, and on the homeland of the Métis Nation. We respect the treaties that were made on these territories. We acknowledge the harms and mistakes of the past, and we dedicate ourselves to moving forward in partnership with Indigenous communities in a spirit of reconciliation and collaboration. My name is Dr. Stephen Kirkland. I'm an Associate Dean of the Faculty of Graduate Studies, and I'll be your MC for this event. 3MT is a research communication competition that challenges students to consolidate their ideas and research so that they can be presented concisely to a non-specialist audience. We believe this competition provides career building opportunities for our grad students, celebrates and promotes the research done at the University of Manitoba, and connects our student researchers with the community. We're proud to be associated with such an inspiring group of graduate researchers who are here today to share their work with all of you. I'm also certain that you'll come away enthused by the work that's being done by these exceptional students. 72 graduate students submitted video applications to be part of this competition. Their submissions were reviewed by assessors from different faculties at the university, and a final grouping of 35 were selected for 3 heats. 10 challengers today will compete today from various faculties. The top 3 from this heat will each be awarded a $200 prize and will advance to the final competition to be held virtually online on the 7th of April at 7 p.m. And of course, you're all invited to tune in and watch that event. Our judges today are Mr. Grant Suderman. Grant is the principal of the strategy consulting firm Verge Strategy Group. His practice with Verge is primarily focused on strategic planning, organizational development, and sales force effectiveness. He has considerable expertise in helping organizations develop strategy, people, and implementing a strong, repeatable sales cadence. He retired as Vice President of Advertising, Sales, Creative, and Marketing at the Winnipeg Free Press in late 2019. Currently, he teaches the retail management course at Asper and coaches the JDC strategy team, as well as mentors with Futurepreneur Canada. He is a member of the Board of Trustees for Manitoba Opera and volunteers his time with Wrench, a community bicycling organization that aims to provide bicycles to those that are less well off. He graduated from the U of M Faculty of Administrative Studies with a BCom honors with a major in marketing. Started as a sales rep and within a few years was director of marketing at Eli Lilly in Toronto. From there, he joined GlaxoSmithKline as a sales director for the Manitoba, Saskatchewan, and Alberta, and where he oversaw the top sales region in Canada with a team of 95 that produced annual sales in excess of $150 million. Our next judge is Dr. Alison Abra. Alison Abra is a warden and vice chancellor of St. John's College. She is an historian of modern Britain, and her research focuses on women and gender, popular culture, and emotion in the era of the 2 world wars. Welcome, Alison. Our final judge is Dr. Brian Rice. Brian holds the position of Indigenous land-based educator in the Department of Kinesiology and Rec Management at the University of Manitoba. He is an enrolled member of the Indigenous Mohawk Nation at Kanawake, Quebec, Canada. He has published 3 books and various chapters and articles on Indigenous issues, history, and culture. So welcome to all of the judges, and thank you for being with us today to choose the 3 finalists who will advance to the 3MT final event competition. Our judges will be assessing our competitors' presentations on the basis of comprehension, engagement, and communication. They have been provided detailed criteria for their evaluations. Each competitor will have only a single overhead slide and 3 minutes for their presentation. The timer will begin when the presenter begins speaking. We will begin with the first 5 challengers and take a short 3-minute break, and then finish the competition with the last 5 challengers. After all the challengers have completed their presentations, the judges will be allowed 10 minutes to tabulate their results and choose the top 3 challengers that will be moving forward to the final 3MT competition at the University of Manitoba. We welcome you to tag the @University of Manitoba on Facebook and Instagram, and @Faculty of Graduate Studies #3MT on Twitter when posting on social media today about this event. It's time to start the competition. Our first challenger today is Taryn Farhana from the Department of Microbiology. Taryn's thesis title is Investigating Susceptibility of IWG Progenies Against 2 Notorious Fungal Pathogens and Exploring Interspecific Interactions of this fungal species. Taryn, please start speaking when you're ready, and the timer will start the countdown.
We all must be quite sick of talking about sicknesses and pathogens that cause them these days, but we can't quite escape them, can we? And for a change, let's talk about how pathogens' life can get as difficult sometimes. We know that microorganisms, either beneficial or pathogenic, are almost everywhere. And like any other organism, these microorganisms interact with each other, and these interactions can actually affect their role in nature. And as I study plant pathogenic microorganisms, I'm going to talk about plant from that perspective. So multiple plant pathogenic microorganisms can inhabit a single host plant as a single host plant at the same time in nature. And how these microorganisms interact with each other, that can actually alter how they cause the diseases, and eventually that can actually impact their disease management practices. So it is really important to study not only plant pathogen interactions, but also pathogen-pathogen interactions. And my study focuses on, for my study, I included 2 agriculturally important microorganisms and microorganisms in the spikelets of my selected host plant and studied their disease development. The first microorganism I'm going to talk about here is Fusarium graminearum, which is one of the most devastating plant pathogens out there. It causes Fusarium head blight and several other diseases to several important cereal crops. And it is one of the most major pathogens, not only because it infects the grains, but also because it produces mycotoxins in the infected grains, which are actually toxic to humans. And the next pathogen that I included in my co-inoculation study is Claviceps purpurea. Claviceps purpurea has almost the same host range as Fusarium graminearum, and the disease it causes is called ergot. And Claviceps purpurea actually is a very host-specific, organ-specific microorganism. It can only infect the ovaries of flowering plants. And when it does, instead of getting a healthy kernel, we're going to get only an ergot kernel, which is blackish structure filled with fungal bodies. And from my study, what I've seen is that when Claviceps present and Fusarium present in the same space at the same time, the Fusarium head blight progression can actually restrict to a certain level. And in case of Claviceps interaction, there was no successful infection in case of Claviceps interaction at all. And so the important question that you need to answer now is that how Fusarium mycotoxin produced by Fusarium graminearum when they get affected by the Claviceps or not. And if they do, could Fusarium produce even higher level of mycotoxins in a highly competitive environment? So we'll have the answers shortly, so stay tuned. Thank you.
Thank you, Taryn. Next up is Chitra Sivakumar from the Department of Biosystems Engineering. Chitra's thesis title is Non-Destructive Analysis of Pulse Flowers.
Congratulations on completing the fall term successfully. It's time to celebrate by baking a cake. Now I go to the store, buy all the necessary ingredients and bake my favorite cake. But something doesn't turn out quite right. When I taste it, it is not satisfying or something that I would be proud to present to my friends at a party. Suddenly, I remember that I used general purpose flour rather than cake flour. Checking the recipe again, I realized the reason why my cake won't win any prizes. You may be surprised to know that the taste and nutritional value of our foods does not just depend on the raw ingredients, but also on how flour is milled. How can we make the flour more guilt-free, more sustainable, more responsible, more nutritious? Incorporating low-cost protein sources like pulse flours, such as milled beans, peas, lentils, that are capable of nitrifying and restoring soil into popular foods such as breads, cookies, and pastas will increase the nutritional value of such foods. And guess what's more? It is also a step towards building a hunger-free world. Imagine having a chocolatey pulse cake. Sounds yummy, eh? It would be both healthy and delicious. But imagine if, when you're enjoying your cake, suddenly you find a piece of dough. This could be a result of uneven size of the flour particles, starch damage, and protein quality of the flours. Interestingly, most of these parameters depend on the milling method. Milling a pulse flour with specific requirements is not an easy task. Unlike wheat flours, there are no well-defined standard milling processes that exist for pulse flours. Also, blending pulse flours from different milling fractions changes the overall chemical composition, which in turn affect their functional and nutritional properties. Therefore, in my thesis, 4 types of Canadian-grown pulses are milled using a roller mill. The flours are blended into various streams to study their microstructure using non-destructive imaging techniques. Specifically, I will look at the flours microscopically to see the structural difference between starch and protein bodies. This study will be important to define pulse flour mill settings so that the flour with required specifications can be created. These specifications are essential for food processors to create designer pulse flour blends for more wholesome and nourishing foods. Consumers can be provided with affordable, fulfilling food with excellent texture and mouthfeel for a hunger-free sound sleep. Now, we all know that for every baked good and confection, there is a specific flour that would best suit it. Given the option, we might all choose a healthier baking option that will salivate many palates. Have your cake and eat it too. Thank you.
Thank you, Chitra. Our third challenger today is Amanda Slagerman from the Department of Kinesiology and Recreation Management. Her thesis title is False Assumptions During Lifting, a Risky Business.
Have you ever picked up a box that was heavier or lighter than you expected, causing you to jerk forwards or backwards? Maybe you even had to take a step to keep from falling. Unexpected changes in weight can be scary, and rightfully so. Situations like this, often found in the workplace, can lead to low back injuries that persist for years or even a lifetime. In fact, over 80% of people will experience a low back injury at some point in their life, with 2/3 of those injuries taking place at work. Low back pain costs upwards of $28 billion annually just in loss of workers' productivity and compensation. But the cost of low back pain goes beyond the financial. Those of us who've experienced chronic pain or know someone who has, we understand the emotional burden that comes with living in pain and ultimately the decreased quality of life. So what can we do to prevent injuries like this in the workplace? Well, we first need to understand what's happening to our body, which is where my research comes in. I'm exploring how our expectations about a load can influence the way we lift, including how we activate our muscles, how fast we lift, and ultimately, what happens when those assumptions are wrong. I'm recruiting healthy adults to do a simple task of lifting 10 identical boxes. Well, not quite. Although the boxes may look the same, their weight will not be, and participants will be unaware that there are 2 boxes, one significantly lighter and one significantly heavier than the rest. Using a variety of research equipment, I will be measuring the timing and amount of muscle activity in the abdomen and the back, while also measuring the movements of the trunk, including how fast and how smoothly they lift. I will be able to see how unexpected increases and decreases in load can significantly change the way our muscles are able to react and how our body moves, so that we can take the appropriate steps for reducing the risk of low back injury, whether it's through improved lifting guidelines, or the design of innovative wearable technology that can help control and support the back. The prevention of low back injuries like this in the workplace is important for reducing the economic strain of low back pain on industry, our healthcare system, and the individual, while ensuring that workers are able to maintain a good quality of life as they work towards retirement. Thank you for listening.
Thank you, Amanda. Up next is Janelle Francis from the Department of Microbiology. Her thesis title is Waterways or Rivers as Vehicles of Enteric Viruses.
After 6 months of minus 30-degree weather, you and your loved ones finally get to enjoy a beach on a summer day. In preparations for the long-awaited beach day, you ensure to pack all the essential items, such as sunscreen, snacks, water bottle, and swimsuits. You prepare lunch for the kids and get everyone ready into the car. Once you get there, you find the perfect spot with just enough shade. You set up your umbrellas and beach chairs as you watch your kids run off into the water. At this moment, all is safe in your mind. But as reported by the Government of Canada, waterborne enteric viral outbreaks have occurred in groundwaters of Alberta and Ontario at 11% positive sample frequency in 2013. Some viral outbreaks also go unperceived or just aren't reported. Now, I probably worried you and left you wondering what might be the cause. The truth is, traditional methods that monitor the microbial quality of our wastewater focus only on the detection of fecal bacteria. This is because fecal bacteria is the current gold standard of microbial aquatic health. However, this can also be partial because the results of a negative fecal bacteria test may disregard the presence of other significant types of waterborne pathogens, such as viruses. My research project addresses this shortfall in microbial detection. We hypothesized that enteric viruses may be used as one form of an alternative proxy of anthropogenic activities in urban aquatic environments. Skimmed milk flocculation, a simple yet reliable technique used in the lab, is able to concentrate viruses from aquatic samples. As shown on my slide, numbered 1 through 11 are 11 aquatic samples that I collected along the shores of the Red and Assiniboine rivers of Winnipeg. Through sequencing, I've been able to determine the taxonomic and functionalities of viruses present. As shown in the pie charts, we discovered that Cyphloviridae, Podoviridae, and Myoviridae are top families of pathogenic DNA-enteric viruses, while Totoviridae and Picobinoviridae are top families of RNA-enteric viruses that are all most abundant in our local rivers. When compared to the UID gene in E. coli, we discovered that enteric viruses were at least 1 to 2 orders of magnitude higher than the E. coli marker. But this is just the beginning. These findings will encourage continuous screening of additional enteric viruses over longer sampling event periods that may hopefully lead to the development of novel viral markers of fecal contamination. Thank you.
Thank you, Janelle. Up next is challenger number 5, who will be followed by a 3-minute break to allow the judges to finalize notes and comments on their scorecards. Challenger number 5 is Caitlin Weissgerber from the Department of Biological Sciences. The thesis title is Investigating Spontaneous Autopolyploidy in Wild and Hatchery Lake Sturgeon.
In Manitoba, lake sturgeon are a historical species, having evolved 184 million years ago and being significant to our Indigenous communities socially and culturally. Unfortunately, commercial exploitation in the early 1900s decimated sturgeon populations, and while conservation efforts have been extensive, they have yet to fully bounce back. I do believe that as our knowledge of the species increases, so too will its population sizes. So in the spirit of increasing knowledge, I want to tell you a little bit of what we know and then how my research will contribute. Lake sturgeon naturally have 8 pairs of chromosomes, meaning that they are octoploid or 8N. During fertilization, however, if the female egg fails to divide its chromosomes properly, the offspring will receive additional copies, making it a 12N. This is what's called spontaneous autopolyploidy, and it happens more often if physical shock or egg aging occur during fertilization. Egg aging is just when the female releases her egg, but the male hasn't got there to fertilize it yet. Both physical shock and egg aging occur more often in fish hatcheries than they do in the wild. And since our 12N offspring is fertile, it can produce or reproduce with the 8N individuals and results in progeny that have 10 chromosomes now. And it's these 10N individuals that are really problematic because they exhibit defects which inhibit their ability to survive. So if even one 12N individual produces progeny, there will be thousands of offspring that are 10N and have those defects. My research will evaluate and compare the ploidies of wild versus hatchery populations of sturgeon, where I'm predicting to find more 10N and 12N individuals in the hatchery because of that increased chance of disturbance during fertilization. The ploidy of the fish will be determined with a blood sample where blood cell size is indicative of ploidy because more chromosomes make the cell larger. For the purpose of conserving this historic species and maintaining the genetic integrity of the population, we really do need to have an inclusive understanding of factors affecting hatchery success. You can think of this research as a quality control, providing hatchery managers with the necessary feedback to ensure that future generations have stable sturgeon populations, much like their ancestors did. Thank you.
Thank you, Caitlin. We'll now take a short break to allow the judges to total their scores so far and make notes on their score sheets. This is also a time for viewers to take a stretch, refill their snacks or beverages, and we will return in 3 minutes. Welcome back. We will continue with the 3-minute thesis competition, heat number 1 with challenger number 6. Chado Patty Aya is in the Department of Physiology and Pathophysiology, and Sukta's thesis title is Old Drugs, New Purpose.
Today, I'm going to tell you about 2 patients. And after hearing about their health problems, you might think that the problems are very different from one another. But my project will show that there might be a common treatment. So imagine a scenario in which a lady suffering from high blood pressure goes to visit a doctor, and the doctor immediately prescribes her the commonly used drugs against high blood pressure, that is, angiotensin converting enzyme inhibitors or ACE inhibitors, or angiotensin II receptor blockers, that is ARBs. Now, both of them are inhibitors of the angiotensin II signaling pathway, as angiotensin II has been seen to be playing a major role in leading to cause high blood pressure. Now imagine another scenario in which a lady showing signs of early stage heart failure goes to visit a doctor. And the doctor, after performing a few tests and examining her, finds out that she may have been exposed to stresses such as like heart attack, valve disease, diabetes, high blood pressure, or even the simple ones such as aging or inflammation that resulted her heart to be transformed from its normal muscular elastic nature into a stiff brick, as I've shown in my slide. This is cardiac fibrosis. which in later stages can lead to causing heart failure and death. Now the doctor is at crossroads because although he knows that she is suffering from cardiac fibrosis, but currently there are no treatment options available for it. So as I mentioned to you in the beginning, through my project, I'm trying to link these two scenarios together and see if the angiotensin II pathway inhibitors, that is ACE inhibitors or ARBs, can block the stiffening of the heart, which is cardiac fibrosis. And while it does that, if a protein called scaraxis has a role to play or not. Because in our lab, we have seen that in response to similar stresses, scaraxis instructs the proteins that form the structural framework of the heart to be overproduced, resulting in the heart to become very stiff. So through my project, I'm aiming at providing not one, but two treatment options. First, the angiotensin II pathway inhibitors, and second, downregulation of scaraxis for helping the millions of people, primarily women, suffering from cardiac fibrosis and preventing their transition towards heart failure and death. Thank you so much.
Thank you, Sikta. Up next is Jana Drummond from the Department of Biological Sciences. Jana's thesis title is Investigating a Unique Organ in Ancient Fish, the Spiral Valve.
Imagine being able to travel back in time hundreds of millions of years ago to examine animals living amongst dinosaurs. With my project, you don't need to imagine it. It's a reality. I study lake sturgeon and Pacific spiny dogfish, two species considered to be ancient fish. Now, before we delve further into understanding the unique aspects of these animals, it's important to understand how vertebrates, or animals like me and you that have a spinal cord, acquire energy. You all have heard of carbon. It's in our pencil leads, it's in our bodies, it's in the air around us right now. Carbon is considered to be the building block of life due to its ability to form various molecular structures, and vertebrates need to consume carbon to derive their energy. This is done through the consumption of carbohydrates, which are broken down into glucose. Glucose is a very important energy source. In fact, utilizing the energy stored in glucose is thought to be one of the most ancient metabolic pathways. However, we currently know very little about glucose acquisition in ancient fish. Now, why should we study lake sturgeon and spiny dogfish? Well, there's a bunch of reasons. However, of particular importance to their gastrointestinal tract is they have a unique organ called the spiral valve. If you look at the spiral valve, you can see that it has lots of folds. These folds are thought to increase the surface area available for nutrient absorption. In fact, the spiral valve is currently thought to be the primary area of nutrient absorption in these animals. However, this is just based off of looking at its structure. We need more evidence to back this up, and that is where my project comes into play. My project aims to increase our understanding of the spiral valve by looking at its role in obtaining the important energy source glucose. The first step of my project is to determine where along the digestive tract carbohydrates are broken down into glucose by analyzing digestive enzymes. So carbohydrates are too big on their own to be taken up by gut cells. Digestive enzymes are what break up carbohydrates into smaller molecules like glucose that can easily be taken up by gut cells. The second step of my project is to determine where along the digestive tract glucose is primarily absorbed. So due to the structure of glucose, it is unable to enter gut cells without help from glucose transporters. Therefore, I will also determine where glucose transporters are primarily located in these organisms. So by comparing the role of the spiral valve in two ancient fish species, it can help us understand an organ we know very little about that has presumably been around for hundreds of millions of years. In order to determine how some of the most ancient living vertebrates are able to obtain an energy source involved in one of the most ancient metabolic pathways, I don't need a time machine. I can do it right now. Thank you.
Thank you, Jana. Challenger number 8 is Alika Li from Pharmacy. Alika's thesis title is Anti-Seizure Treatments During Pregnancy and Neonatal Birth Outcomes.
Imagine a pregnant woman well into her third trimester walking down the street, and suddenly she has a seizure. Just imagining the scenario is scary as it can lead to unwanted consequences. For example, the infant can have hypoxia, That is, decreased supply of oxygen to the infant's brain that can lead to long-term adverse outcomes in infant health. Or the woman can have a fall due to the seizure that can lead to trauma in both mother and child, which ultimately led to death. Therefore, it is essential to treat these seizures. But anti-seizure medication, which are used to treat these seizures, can also cause adverse outcomes in infants when exposed during pregnancy, like malformations or preterm birth. Therefore, mothers always ask, should I take the drug or should I not take the drug? Is this medication safe for my baby? And physicians are always in search for that anti-seizure medication with the best seizure control. My research will help answer this conundrum. I'll be doing 2 projects as part of my thesis. And my first project, I'm currently doing a systematic review and meta-analysis where I will summarize all the published evidence up until this point on the association of anti-seizure medications and various birth weight outcomes. We found that women who are exposed to anti-seizure medications have 55% increased risk of having an infant with low birth weight than compared with unexposed women, showing that, yes, indeed, there is a correlation between anti-seizure exposure and low birth weight. Going forward, I will conduct a pan-Canadian cohort study with administrative health databases from Manitoba, Saskatchewan, Alberta, and Quebec, which will include 1.5 million pregnancies, where again, we will study the association of anti-seizure medication and neonatal birth outcomes, including various birth weight outcomes, as well as preterm birth. The knowledge generated from my research projects will help in pre-counseling women before they get pregnant, and also help ensuring pregnant women to take their medications and to avoid any seizures, which can be avoidable, and also indeed avoiding unforeseen consequences. This results knowledge will also help physicians choose the best anti-seizure medication with the best seizure control, as well as the least risk of neonatal adverse outcomes, therefore improving the lives of infants, not only in Canada, but also worldwide. Thank you.
Thank you, Alika. Up next is Badriya Mubashira from the Department of Microbiology. Badriya's thesis title is Metabolic Model, a Hypothesis Generator.
You have a family of 9 members, but you have food for only 2. Not a big problem. Now think of these numbers in billions. Tough choice, right? We would have found ourselves in this exact situation if we hadn't found ways to boost our food production to feed this ever-growing global population. So as for our food, we are highly dependent on plants, even though no matter how much we take plants for granted, if plants had a union, they would be the highest paid inhabitants of the planet. And for plants, they need nutrients to grow, and one of which is nitrogen. And we have a lot of nitrogen around us. Even the air we breathe in, that's 80% nitrogen. But the nitrogen present in the air is not in the exact form that plants can use that. So in early 20th century, a German chemist named Fritz Haber found a way to synthesize this usable form of nitrogen from directly air. But this fertilizer, this leads to for formulation of fertilizer that helped us industrialize our agricultural sector. But every good thing comes with their own liabilities. For one, using nitrogen fertilizer leads to emission of nitrous oxide, which is 300 times more harmful than greenhouse gas carbon dioxide. And also, fertilizer finds their way from fields to water bodies that actually interferes with their aquatic ecosystem that in turn, in the long run, affects us. So is there any other sustainable alternative? Up our sleeves, we have a friend called bacteria. There are bacteria, for example, rhizobia, that maintains some sort of friendship with some legume plants, such as peas, beans, where the bacteria says to plants, I will pull nitrogen from air and just give it to you. In return, just give me food and shelter so that I can survive. My work is to make a model or prototype of this rhizobial metabolism, which is the chemical engine where all the secret lies. So we all know why prototypes are made for. For example, a prototype of a rocket or a phone or a car is made so that we can try out new features before we go from design to manufacture. So this metabolic model will help us in the same way by generating hypotheses so that we can know more about what goes on in these bacteria and how they maintain this mutualistic relationship and exploit those information in our favor. And in future, we hope to provide our farmers with a sustainable alternative so that the use of chemical fertilizer can be decreased, or even better, if they don't have to use fertilizer at all. And also, most importantly, maintaining the same level or more crop production.
Thank you, Badriya. Our final challenger today in the first heat of the 3MT competition is Chris Voth from the Department of Kinesiology and Recreation Management. Chris's thesis title is Shining a Light on the Unseen Athletes.
When I was growing up and started becoming aware of my sexuality, I thought that I was going to have to stay in the closet forever, especially if I wanted to be a professional athlete. At that time, athletes were only coming out after their careers had ended. However, I wanted to be the role model that I never had, so I came out as Canada's first active openly gay national team athlete. There were a lot of unknowns, especially if my coming out would hinder my ability to play volleyball professionally overseas. Unfortunately, that fear turned into reality when a team cited that as their reason for not hiring me. My agent told me that this was not an anomaly, but that they were the first team to say it so bluntly. Sport is currently showcasing the most out athletes than ever before. However, there's a distinct pattern among those coming out. By differentiating between men and women and individual versus team sport athletes, I was able to uncover a major lack of representation. It wasn't until Tokyo 2020 where we saw the first gay male team sport athlete competing on the Olympic stage. Last year, we also saw the first gay male to come out while playing in one of the major North American team sports. The NHL hasn't had an athlete come out even after their career, though. Currently, most of the research surrounding LGBTQ+ athletes covers the coming out of high school, college, university, individual sport athletes, and thankfully largely is all positive. But due to the lack of representation in the professional realm, it's apparent that the accepting culture is not universal. I know that my professional career was greatly impacted by my sexuality, but there's currently no research on this topic yet. One of the major roadblocks for the research is having access to out or closet athletes, but this is where I'm in a unique position to conduct this original study. Through individual interviews with gay team sport professional athletes from around the world, I have been exploring their experiences and finding common themes. My data collection has not concluded yet, but so far there have been some interesting potential results. If publicly out, these professional athletes are celebrities in their sport, especially from their home regions, and are ambassadors of the gay community. Because of the pressure to be a positive role model, many spoke to the reasons they underreported the homophobia that they experienced. Some athletes also indicated that they didn't want a reputation for complaining about their teams because of the fear that future teams will want to hire them. By getting the chance to talk about their experiences, I've also seen areas to improve the team culture and make the environment more welcoming. My goal is to share all of these findings so that teammates, coaches, sport organizations, and fans can all be a part of making sport more inclusive for gay athletes. Through making these changes, more gay athletes might feel comfortable coming out or to play sport that they previously felt excluded from. As seen with racism and sexism, sport can act as a catalyst for sparking societal change. We are approaching another tipping point for inclusion, and I hope that shining a light on the unseen athletes can tip the scales. Thank you.
Thank you, Chris. This completes the challenger presentations for this morning. Thank you to all our challengers who presented today. These were amazing presentations on very diverse research topics. A virtual round of applause to all of you. Judges will now meet virtually in a breakout room to total their score sheets and to determine the winners. The judges' results will be announced live in just a few minutes, so please don't go away. While we wait for the judges' results, there will be a short slideshow introducing you to some of the university's past 3MT winners. Enjoy the video. Welcome back, everyone. The judges have made their decisions and they want to express that it was not an easy task as the challengers were outstanding and their presentations were very well done. They did want to mention that they were very impressed with the content of the presentations and how well each challenger did, especially in this online format in which the audience is invisible, unfortunately. Presenting virtually is a skill that has been made a necessity in the last 2 years, and every challenger should be proud of their presentations. Challengers that will be advancing to the finals on April 7th and who will each receive a $200 prize are, in no particular order, Alika Li, Janelle Francis, and Chitra Sivakumar. Congratulations to the finalists. There will be a wild card, yes, applause all. There will be a wild card selection announced after the final 3MT heat. This wild card will be selected by a committee that will attend all 3 of the 3MT heats and make its choice from all of the competitors from the 3 heats. The wild card selection will also advance to the finals, but will not be awarded $200. The wildcard selection will be notified by email early next week after all 3 heats are completed. This has been a wonderful session of diverse ideas and exciting research. Thank you to the challengers for sharing your research with us. I would also like to thank our technical team of David and Teddy, who were instrumental in facilitating the live event today. And thank you to the students, their advisors, their friends and family for the support to make the 10th annual 3MT heat a success. And also thanks to our panel of terrific judges, Mr. Grant Suderman, Dr. Allison Abra, and Dr. Brian Rice. One final reminder that the University of Manitoba's 3MT final competition is on April 7th, virtually at 7 p.m. Everyone is welcome to attend. The link will be on the U of M Faculty of Graduate Studies webpage at umanitoba.ca/3MT. Thank you all for attending wherever you might be today. On behalf of the Faculty of Grad Studies, have a good rest of your day.